In this video, I'm going to go over some of the activators and inhibitors and allosteric effectors of the different enzymes in the glycolysis and gluconeogenesis pathway. So these are really only going to affect the enzymes that are the inverse of one another, so the gluconeogenesis counterpart of the glycolysis enzymes. So we're going to go through what inhibits each one, what activates each one, if there's any covalent control, and we're just going to take it step by step. So starting off with the very first enzyme, hexokinase. Now, hexokinase is inhibited by glucose 6-phosphate. Glucose 6-phosphate inhibits. Now, this makes sense because if there's a lot of glucose 6-phosphate, then you're not going to want hexokinase to work that well because it turns glucose into glucose 6-phosphate, so this is a kind of negative feedback control. Now, the opposite is for glucose 6-phosphatase. Here, G6P activates it. Because if there's a lot of this one substrate, it's going to want to maintain balance and try to make more glucose. It makes sense that glucose 6-phosphate would have opposing effects of both of these enzymes because you would not want glucose 6-phosphate to have a similar effect because then it's just going to be the same cycle over and over again. If glucose 6-phosphate both activates glucose 6-phosphatase and hexokinase, you're just going to get a cycle of glucose 6-phosphate over and over and over again. So this is why they have completely different effects on each of these enzymes, which you will see is a pattern as we move forward. Next, we're going to talk about phosphofructokinase. So this enzyme, its inhibitors include high ATP and citrate. Now this makes sense because glycolysis, the whole purpose is to produce ATP. So if there's a lot of ATP, then we're not going to want these enzymes to be working to produce more ATP if it's not necessary. Now its activators are activated by the opposite, so low ATP or high AMP, or something called fructose 2,6-bisphosphate. And we'll talk about this here in a second. But first, let's scooch over to its counter enzyme from gluconeogenesis, fructose 1,6-bisphosphatase, and its feedback control is exactly the opposite. So its inhibitors are AMP and fructose 2,6-bisphosphate, and its activators are ATP. Now, what is this fructose 2,6-bisphosphate that I keep talking about? Let's go a little bit below so we have some more room. Fructose 2,6-bisphosphate is synthesized by phosphofructokinase 2. It is degraded by FBPase. And protein kinase can either phosphorylate or unphosphorylate it, and different kinds of phosphorylation makes it have different effects. So let's start with the phosphorylated form. The phosphorylated form is fructose 2-phosphate. So in this version, there is less fructose 2,6-bisphosphate. So in order for there to be less of that, it is going to be activated by FBPase, and then PFK2 is going to be inactivated. So this all leads to less fructose 2,6-bisphosphate. And this basically means that glycolysis is inhibited, and gluconeogenesis is going to be favored. And this is when we have low blood sugar. Now let's switch to the unphosphorylated form. So the unphosphorylated form is going to be fructose 2,6-bisphosphate. And so everything here is opposite. So FBPase is inhibited, PFK2 is activated, and this all means that there's more F26BP. This stimulates glycolysis and inhibits gluconeogenesis. And this is in response to high blood sugar. So that is fructose 1,6-bisphosphate. It's just an important thing to remember when it comes to activating and inactivating phosphofructokinase and fructose 1,6-bisphosphate. So as you can see, we said fructose 1,6-bisphosphate activates phosphofructokinase, which, as we mentioned down here, stimulates glucose. So it's going to stimulate the glucose enzyme, and it's going to inhibit the gluconeogenesis enzyme. Moving on, we are going to go all the way down to pyruvate kinase. So pyruvate kinase is inhibited by ATP and alanine. And this is allosteric feedback, but there's also covalent feedback. And this is phosphorylation. So if it is phosphorylated, this makes the enzyme less active. And this happens when there is low blood sugar because we don't want to perform glycolysis. So going to its counterpart enzyme from gluconeogenesis, pyruvate carboxylase is activated by acetyl-CoA. Okay, and that is pretty much all of the allosteric and kind of activators and inhibitors of gluconeogenesis and glycolysis. And as you notice, it really only happens to the enzymes who have counterparts for glycolysis and gluconeogenesis, so only those 3 steps. So it's important to know what kind of environments will favor glycolysis and what kind of environments will favor gluconeogenesis. And then from there, you can kind of deduct, okay, what enzymes will it favor based on which pathway it's in. But that is all for this video. The notes will be on the link tree, and in the next video I will go over some of the activators and inhibitors for the other pathways.